Thank you for joining our Debbie's Dream Foundation Curing Stomach Cancer webinar series. Today's webinar is titled The Role of Bacteria in the Development of Stomach Cancer, Risk Factors and Therapeutic Options for the Prevention of Disease. And this is the 11th in a series of 12 monthly webinars. I am Mary Margaret Kilmeyer, and I'll be moderating today's webinar. I am the Programs Director for Debbie's Dream Foundation Curing Stomach Cancer, and I'm a licensed marriage and family therapist. My clinical and research background is focused on working as part of a collaborative team with doctors, patients, their families, and the members of a healthcare team. We would like to thank our title sponsors for this webinar series, Boston Biomedical and EMD Serono, as well as our platinum sponsor, Lilly Oncology, who have provided the funding to make this webinar series possible. You will be able to ask questions during this presentation. You can type your question into the white text box that appears on your screen. We will address questions at the conclusion of the presentation. And in addition, the recording of this webinar will be accessible on our website in the lecture library. First, I will share information about stomach cancer, our president and founder, Debbie Zellman, and the organization. And then we will hear a presentation on the role of bacteria in the development of stomach cancer from Dr. Richard Peek of Vanderbilt University. And then we will conclude the presentation with a question and answer session. Some of you may be familiar with facts and statistics about stomach cancer, and we are continually updating them as new information becomes available. In 2017, it's estimated that more than 28,000 Americans will be diagnosed with stomach cancer, and nearly 950,000 diagnosed worldwide. In the U.S., more than 11,000 individuals will die from this illness. 80% of all stomach cancers in this country are also diagnosed at stage 4, when the 5-year survival rate is only 5%. And incidence rates in younger populations continues to increase while the expected frequency has been on the decline. And still, very many know little about this deadly disease. Pictured here is the president and founder of Debbie's Dream Foundation Curing Stomach Cancer. Debbie was diagnosed with stage 4 stomach cancer in April of 2008. She was a practicing attorney and mother of 3, and she had no risk factors for stomach cancer, and her symptoms were very vague at the time. When she was diagnosed, she was told that her chance of being alive was only 5 years, was only 4%. She endured harsh chemo regimens and targeted treatments and experienced many recurrences over 9.5 years. Unfortunately, Debbie passed away just recently on December 23rd at the age of 50 due to her illness. She dedicated the last 9.5 years to helping others with stomach cancer by raising awareness, funding research, and providing resources and education through this organization. Debbie founded DDF in April of 2009. As an organization, we're a member of several advocacy coalitions, including the Deadliest Cancers Coalition, the Patient Equal Access Coalition, the State Patient Equal Access Coalition, and One Voice Against Cancer. Debbie served for many years as a patient advocate on numerous committees and task forces to help raise the profile and voice of stomach cancer patients. And as an organization, DDF will continue her important work to honor her legacy. If you have any questions or would like to send condolences to the organization or the family for Debbie, please email programs@debbiesdream.org. Debbie's Dream Foundation is dedicated to raising awareness about stomach cancer, advancing funding for research, and providing education and support internationally to patients, their families, and caregivers. Our ultimate goal is to make the cure for stomach cancer a reality. You can learn more about our organization by visiting the website at www.debbiesdream.org. In the few short years that DDF has been in existence, we have achieved many great milestones. We have 25 chapters across the U.S., as well as chapters in Canada and in Germany. We have events that are ongoing around the country. Our patient resource education program helps patients and their families, as well as caregivers around the world, by matching them directly with survivors and caregivers by using disease-specific criteria, including stage, biomarker, and location. We host educational webinars like the one you're listening to today, as well as symposia year-round and around the country. Our website contains in-depth information about stomach cancer that can be translated into more than 60 languages. We have also provided $650,000 in research grants, as well as having an open $50,000 research call for research grants currently. We also advocate for stomach cancer research money at Capitol Hill during our annual Advocacy Day, and this is to add stomach cancer to Department of Defense's peer-reviewed cancer research program. And these efforts have resulted in nearly $18 million being awarded to stomach cancer researchers over the last 3 years. Here you can see a snapshot of our website's homepage, including in remembrance of Debbie Zellman, as well as many numerous links to information and support resources that you can access. And here's a listing of some of the upcoming events over the next several months. That includes current webinars. We're also hosting a Night of Laughter in Fort Lauderdale on January 28th. Our annual Capitol Hill Advocacy Day, which will be the 6th of the annual Advocacy Days, will be hosted on February 26th and 27th. This is an incredibly important event to help inform Congress about the importance of research funding for stomach cancer, as it receives the least amount of funding per cancer death of all cancer types. So please consider joining us in Washington. We'll also be hosting our annual Stomach Cancer Education Symposium and webcast on April 21st. And that evening, it will be followed by our 9th annual Dream Makers Gala, both in Fort Lauderdale. To learn about these and other events, please visit our website and click on the tab Events. Debbie's Dream Foundation is headquartered in Plantation, Florida. Our office hours are Monday through Friday, 8 a.m. to 5 p.m. Eastern Time. On this slide are also some important phone numbers and email addresses that you can use to contact our office and our staff. We'll now begin the presentation, made possible in part by our title sponsors, Boston Biomedical and EMD Serono. The topic this month is the role of bacteria in the development of stomach cancer. And our speaker is Dr. Richard Peek of Vanderbilt University. Dr. Peek is the Director of Gastroenterology at Vanderbilt University. He's a professor of medicine, cancer biology, pathology, microbiology, and immunology. He's received numerous honors, awards, and academic appointments, in addition to being named the editor-in-chief of the journal Gastroenterology. We're so thankful for him joining us today. And now, Dr. Peek, I'd like to turn the webinar over to you. Thank you again.
Great. Well, Mary Margaret, I'd like to thank you so much for that very kind invitation. I'd also like to thank Kathleen Lane for inviting me to be able to do this. This is an incredible honor for me. I had the chance to meet Debbie at a conference in Washington, D.C. last year, and I was immediately struck by her passion and her devotion to the treatment of this disease. She's a very powerful advocate, and what she's done with this foundation, I think, will lead efforts in this disease process well into the future. So again, I'd like to extend my sympathy and condolences to everybody who knows Debbie and who has worked at the foundation, but also I think that there's much hope based on what she did. So thank you again for this honor. What I'd like to do this afternoon is just talk a little bit about some research that we've done and other labs have done that have focused on what I consider to be a paradigm shift in the way that we think about cancer. And that's actually to consider the role of bacteria in the development of not only stomach cancer, but also other cancers that arise from areas of inflammation. So can we have the next slide? So I've worked on gastric cancer here at Vanderbilt for 25 years. The person that I trained with actually was one of the pioneers to understanding how bacteria may cause cancer. His name is Martin Blaser. He's currently at NYU, but he started me on this journey that I've enjoyed every step of the way. And so I'll talk a little bit at first about how bacterial infections may cause stomach cancer, and then more importantly at near the end of the talk, I'll talk about the clinical, diagnostic, and therapeutic regimens that we have available to eliminate this risk. Next slide. As Mary Margaret mentioned, gastric cancer is a very important disease, especially worldwide. Currently, there are almost 1 million new cases that are diagnosed every year, and currently it's the 3rd leading cause of cancer-related death. And this accounts for over 700,000 deaths per year. In the United States, if you look at the incidence rates of gastric cancer, they've been declining over the last century, but now we're starting to see increases in particular populations, especially immigrants who have migrated from areas in which gastric cancer rates are very high, and also other populations such as Hispanics. They are seeing an increase in the rates of gastric cancer. So it's a very important problem, not only worldwide, but also within the United States. Next slide. As a gastroenterologist, this is an unfortunate lesion that we too frequently diagnose. So this is a picture of the inside of a stomach that I took during an upper endoscopy procedure I did 2 years ago. And what you see at 6 o'clock in the picture is a mass. It's more red than the other parts of the stomach lining. It's very irregular. And when we biopsied this, it bled very easily. And those are all endoscopic characteristics of a malignant mass. And when we looked at the biopsies, this patient unfortunately had a type of gastric cancer that's actually the most frequent type, and that's called intestinal type gastric adenocarcinoma. Next slide. We've known for years, based on expert pathologists, actually the steps that a person will go through histologically in the stomach before they'll develop intestinal type gastric adenocarcinoma. In the upper left is a histologic picture of a normal stomach. There's no inflammation there, no reason for concern. But the steps that are going towards gastric cancer actually progress through a very well-described pattern. The next step in this process is a step that we call non-atrophic gastritis, and that just means that there is a lot of inflammation in the stomach lining. Over a period of decades, people can actually progress from this lesion to what we call atrophic gastritis, in which gastric glands are lost. And it's interesting because many of the cells that are lost in a person that has atrophic gastritis are acid-producing parietal cells. And so these people actually cannot make much acid. And so when a person comes in who's not been treated with any drugs that we use to heal ulcers, such as proton pump inhibitors, and they're not making acid, that's a worrisome sign. And we will use increased scrutiny during endoscopy and try and biopsy areas in the stomach. Individuals can then move on to develop intestinal metaplasia, and then this is a strong risk factor for the development of both dysplasia and gastric cancer. Before we knew about bacteria in the stomach, many of the risk factors that led to gastric cancer were thought to be either genetic or diet-induced. It was known for years through epidemiology studies that high-salt diets or individuals that consumed pickled foods had an increased risk for gastric cancer. But we never knew actually how this happened. Next slide. So several years ago, there really was a landmark discovery that changed the way that we looked at gastric cancer, how we understood gastric cancer, and actually how we can go about preventing this disease. So the picture that you're looking at now is a biopsy of a stomach, and the little black lines that you see within the white area or the gastric lumen are actually bacteria. And this picture was taken probably 15 or 20 years ago, and it shows that the bacteria are very close to the epithelial cells. This is what they use for nutrients. And we now know these bacteria to be called Helicobacter pylori, or H. pylori. Next slide. The individuals that were credited for discovering this bacteria are shown here. It's quite an interesting story. So the person on the left with the mustache is Barry Marshall. The person on the right with the beard is Robin Warren. So Barry Marshall, they're both from Australia. They lived in Perth. Barry Marshall was a gastroenterologist, and he was doing his fellowship at the University of Perth. And he was told that he had to have a research project completed before they would certify him as a attending gastroenterologist. So he went down and he talked to Dr. Warren about a potential research project. And Dr. Warren said, well, you know, I've been noticing a lot that patients who go to the operating room for either ulcer disease surgery or gastric cancer surgery, when I look at the specimens, I'm seeing a lot of bacteria. And pathologists had known this for years, but he was seeing more of the bacteria in the samples from ulcer patients and patients with gastric cancer than from patients who had operations for other reasons. So he said we should, you know, try to understand whether these bacteria are causing these diseases or exerting any kind of role, or whether they're just innocent bystanders or even contaminants. So Dr. Marshall set upon trying to grow these bacteria. They're very difficult to grow. You have to have special culture conditions, anaerobic cultures. And the story goes that on Easter weekend, the whole lab left for a week, and they left their culture plates in the incubator, and they forgot about them. And when they came back a week later, which is much longer than you would normally leave bacterial culture plates in an incubator, they actually found the bacteria growing. It took that long to grow on a blood plate. And from that point on, they were able to culture the bacteria and prove that it was a major cause of ulcer disease and gastric cancer. Next slide. And so for this discovery, Dr. Marshall and Dr. Warren won the Nobel Prize in 2005. And this is a picture showing them in Stockholm after they received the Nobel Prize medals. Unfortunately, Dr. Warren on the left has passed away, but Dr. Marshall is still very active in research on H. pylori, ulcer disease, and gastric cancer. And I've been able to interact with him on several occasions. Next slide. So this is what we see when we look close up in a biopsy of a person that has ulcer disease and is also infected with H. pylori. You got somewhat of a sense of this on the slide I showed previously, but all those black dots that you're seeing in the middle of this histologic picture are inflammatory cells, and they normally are not there in a person who's infected with H. pylori. Next slide. And if we look on higher view, these inflammatory cells are disrupting the whole organization and architecture of the gastric glands. They're infiltrating within this round circular gland. They're disrupting it. And this is not what you see in a normal situation. Next slide. So based on this, and based on the fact that H. pylori now causes inflammation in virtually everybody who's infected, we think that the process of gastric cancer is really initiated by this infection early on. It initiates the progression from normal gastric tissue to non-atrophic gastritis, and it also may exert effects later on in this disease progression. Next slide. So currently, H. pylori is an attributable risk for about 90% of the gastric cancers that are diagnosed worldwide. And therefore, it's the strongest known risk factor for this cancer. Next slide. The problem is that there are billions of people who are infected with H. pylori. And the most recent analysis estimated that there were 4.4 billion people who are infected, and that's over half of the world's population. but only 1 to 3% of people that are infected ever get gastric cancer. Next slide. So this is really, I think, driven investigators to try and understand, from a mechanistic level, how does H. pylori cause cancer, and in what individuals does it exert the most risk? And so when we think about H. pylori-induced gastric cancer, we can break down risk factors into different categories. There's certain bacterial factors that I'll just mention briefly later that increase the risk for gastric cancer. There are human factors that include genetic mutations that can increase the risk, and also there are environmental cofactors such as diet that can also exert an effect on gastric cancer. Next slide. Next slide. Next slide. But what we really need to do as investigators is to not just study all these factors in isolation, but really understand how they interact with each other at a molecular level. And that way we will fully be able to understand how they exert risk on this particular disease. Next slide. So our lab is particularly interested in H. pylori, in differences in H. pylori strains. Specifically, what is different about an H. pylori strain that is isolated from a person with gastric cancer versus a person that just has gastric inflammation and never progresses along the carcinogenic pathway. Next slide. Next slide. So one bacterial factor that was identified very early on is a bacterial protein, and it's called CAG-A. And what I've shown in this schematic is H. pylori, shown in green, is bound very closely to an epithelial cell of the human host. And when H. pylori comes into contact with human epithelial cells, it forms a little needle-like structure, and that's the little appendage that you see touching the epithelial cell. And through this needle-like apparatus, it can inject a protein called CAG-A into actually a human cell. And once it's inside that cell, it can interact and activate many different pathways that have all been shown to be mutated in patients who have gastric cancer. There's other evidence that suggests that if you put this CAG-A protein just by itself in an animal, those animals will get gastric cancer. So people have labeled the CAG-A protein as an actual oncoprotein, in that once it's inside a human cell, it can actually transform that cell to become malignant. Next slide. And this is just one example of what that bacterial protein can do to a human cell. So what we've done here is we've put a green tag on the CAG-A protein and then put that protein inside one cell. And so the cell that's labeled, that's showing green, is a cell that harbors CAG-A inside. And you can see that the shape of that cell is much different than the cells that surround it. The cells that surround it do not have any CAG-A in them. But the cell that does, it's more long, it's spindle shape. We have movies that we've shown in which that cell moves much faster through the other cells, and all those are properties of cancer cells. So again, once CAG-A gets inside the host cell, it can exert many different effects that seem to predispose to cancer. Next slide. Well, there are also other factors, as I mentioned, that seem to increase the risk that an H. pylori infected person may develop gastric cancer. One of these categories is host genotypes. And there have been many studies now that have shown that individuals that have mutations in specific immune response genes, one of which is called IL-1 beta, one of which is called TNF alpha, all have an increased risk of developing gastric cancer. But that risk is only there if a person is also infected with H. pylori. So if a person has these mutations and they're not infected, then they do not have any increased risk for disease. So again, it's an interaction between the presence of the bacteria and the genetic background of the host. Next slide. It also appears that where the infection in the stomach is actually can make a difference as to whether a person will be completely asymptomatic or develop gastric cancer or perhaps ulcer disease. Next slide. So this diagram depicts the main outcomes of H. pylori infection in humans. I'll state from the outset that most persons, that's 90% of individuals who are infected with H. pylori, will not develop any symptoms at all that are attributable to this infection. They have mild gastric inflammation. It involves the whole stomach, and this is shown on the upper picture of the stomach. And really, those individuals never have a problem with this infection for their entire lifetime. In the lower left is sort of a schematic showing what kind of pattern of inflammation and infection a person will have if they develop ulcers following infection. And in these individuals, the infection mainly involves the lower part of the stomach, which is called the gastric antrum. And what that does is it allows the upper part of the stomach, which is the gastric body, and that's where acid is produced, that allows that to continue to make high levels of acid. And because acid is important in causing ulcers, persons who have this pattern of gastritis are more likely to develop a duodenal ulcer compared to a person who just has inflammation throughout the whole stomach. In contrast to these two, the stomach picture in the lower right shows the pattern of inflammation a person develops who's more predisposed to develop gastric cancer. And in these individuals, the inflammation involves really the body or the middle part of the stomach. And because inflammation is there, it can inhibit acid production. And this goes back to what I was talking about earlier, where acid levels generally are very, very low in persons that are at risk for developing gastric cancer. Next slide. We've also noticed in the past 2 decades that treatment with certain drugs can change the pattern of gastritis and also thus change the disease that persons who are infected with H. pylori may develop. And that is that, we have drugs that are proton pump inhibitors, such as omeprazole or pantoprazole. They're very effective at shutting off acid production in the stomach. However, if a person is infected with H. pylori and you treat them with a proton pump inhibitor, then frequently they will develop a pattern of gastritis that is more of a risk factor for gastric cancer. So we can convert a person who may develop duodenal ulcers to one that may develop gastric cancer simply by treating them with a proton pump inhibitor. Next slide. The other risk factor that I just wanted to briefly mention today is environmental cofactors. Next slide. And the one that we're interested in here really has to do with iron, and that is iron deficiency. Next slide. So we've known for years that bacteria are very dependent on iron levels. Without iron, many bacteria cannot survive, and they actually have developed virulence factors to be able to acquire iron from their host to allow them to survive for prolonged periods of time. We've also known for a long time that H. pylori infection is associated with iron deficiency, and that iron deficiency is also associated with a high incidence of cancer of the stomach. Next slide. So we've done studies in our lab really to determine the role of iron deficiency in H. pylori-induced gastric carcinogenesis. Next slide. So we have an animal model that we developed here in which we can infect Mongolian gerbils with H. pylori, and they'll develop gastric cancer within 8 to 12 weeks. And we did a simple study in which we treated gerbils with a normal diet, shown in red, or an iron-deficient diet, shown in yellow, and then infected them with H. pylori. Next slide. And what we found was that there was twice as much gastric cancer, shown in red, and twice as much gastric dysplasia, shown in yellow, if the animals were iron deficient when we infected them with H. pylori. Next slide. So how does this happen? Well, we went back to those studies that focused on that protein called CAG-A to understand whether iron levels may regulate the either the needle apparatus that delivers CAG-A or CAG-A itself. Next slide. So what we did was we took H. pylori strains, and this is an electron microscopy picture from animals that had normal iron levels. We added them to cells, and those yellow arrows actually are showing the needles that are penetrating the human host cell and delivering that oncoprotein. Next slide. But look at the strain that comes from an iron deficient animal. Many more of these needles are assembled, and so more of that oncoprotein is being delivered into the host cell. So somehow, in the presence of iron deficiency, the CAG island or the CAG needle is formed to inject more CAG-A into the cell. And we think that this is important, especially if a person is infected for decades. Next slide. We also always try to validate what we find in our animal models and in our cell culture system with human populations. And so we have a very strong population of study in Colombia, South America. And what we were able to do is look at the blood concentrations of iron in persons that had gastritis alone or atrophic gastritis or more severe lesion, which is metaplasia and dysplasia. And their iron levels were the lowest if they had more severe disease. Next slide. And finally, we confirmed this because we took H. pylori strains from those patients. And we found that the H. pylori strains that came from patients that were iron deficient, shown in yellow, could induce much more inflammation than if strains came from patients that had normal or high levels of iron. Next slide. So based on this, we've been able to construct what we think is a working model of how all these things interact. As a person over decades progresses from uninfected stomach to gastric adenocarcinoma, their iron levels go down. This increases the virulence of the bacteria, and there's more inflammation as you go along the pathway. And so we think that iron levels may be a way in the future that we could screen for patients that are more at risk for developing gastric cancer. Next slide. So I wanted to talk a little bit now about treatment. So treatment of H. pylori, we use antibiotics. But the first question was, if you treated this infection, can you really reduce the risk of developing gastric cancer after antibiotics are given? Next slide. One of the first trials that looked at this was performed in China. This is sort of the study design. It started in 1995. They did 2 follow-ups of those patients that were treated for H. pylori in 2003 and 2010. Next slide. And they had 3 different treatments that were given. They gave the patients antibiotics for H. pylori. They gave them garlic, or they gave them vitamin C, sort of as an antioxidant. Next slide. And what they found was that indeed, if they treated these patients with antibiotics, as 15 years later, they did have a significantly reduced risk of developing gastric cancer, and the risk reduction was about 40%. Next slide. And when they combined their results with results from the other placebo-controlled double-blind trials, again, when you pool all those results, there is a significant reduction in gastric cancer rates if individuals are treated with antibiotics that target H. pylori. Next slide. So now we know that treatment of this infection can affect disease later on. So what really are the optimal diagnostic and treatment approaches to eradicate H. pylori? Next slide. I think the most important list that we'll talk about really is here. I mentioned earlier that there are over 4 billion people that are infected worldwide. Only a minority of persons who are infected ever develop any disease at all. So this really argues against a global indiscriminate eradication approach. You know, antibiotics have side effects. We're learning a lot more about the microbiome now and the consequences of changing the microbiome after an individual takes antibiotics. There are also some interesting findings now that H. pylori is actually inversely related to other diseases, allergy, eczema, and esophageal cancer. So there are many reasons to argue against sort of an indiscriminate strategy to treat everybody in the world who's infected with H. pylori. But there are high-risk groups. And in this list, I've listed several different categories of people that I would test and I would treat for H. pylori. Obviously, if people who have ulcer disease or people that have a more rare type of gastric cancer called a maltoma, anybody who has a first-degree relative with gastric cancer, I would test and treat. People that have a particular disease called Menetrier's disease, because there's an increased risk of malignancy there. Persons that have to take non-steroidals or aspirin for long periods of time. There's a synergistic relationship between H. pylori and ulcer disease in those patients. Patients who have what we call non-ulcer dyspepsia, and that's nondescript pain that's generally in the middle of the abdomen, but really the only thing that you can find is the presence of H. pylori. Those individuals have shown to be symptomatically improved if they're treated for H. pylori. I mentioned also that when you treat some people with proton pump inhibitors, it can drive them towards more risky colonization patterns. So I would test and treat those individuals. And then obviously people that have more severe lesions across the gastric cancer cascade. If they have H. pylori infection, I would treat them. Next slide. So we can diagnose this infection many different ways. We can use non-invasive tests where someone does not have to have an endoscopy. There's a breath test we can use. You can do a blood test, although this really is not good to confirm cure. And then there's also a stool antigen test. Next slide. If a patient is going to have an endoscopy, we can take biopsies and also diagnose the infection that way. We can do a test called a urease test because H. pylori makes a lot of urease to exist in the stomach. We can also look at it under the microscope to see the presence of the bacteria, and then we can also culture the bacteria from the biopsies. Next slide. We always want to confirm that somebody is cured of H. pylori. And so the best tests that we have for this are, again, a breath test or a stool antigen test. Next slide. So what about treatment? It seems like it would be pretty straightforward. It's a bacterial infection. We have many antibiotics that are effective against bacteria. Why can't we treat this infection with one antibiotic over 3 days like we can treat, for example, a urinary tract infection? Well, the stomach is a different environment. The stomach makes a lot of acid, and many of the antibiotics that we use are inactivated in an acidic environment. When I showed you the slide showing H. pylori deep within the gastric glands, sometimes antibiotics have a hard time penetrating down into those areas and cannot access all the bacteria that are in the stomach. I think that the target that we would shoot for always is about 90% eradication rate. We also know that the most important factor in determining success of treatment is whether or not the H. pylori strain that a person harbors is resistant to a drug called clarithromycin, because clarithromycin is a drug that we use in many of these regimens. And then finally, if we extend the treatment duration out to 14 days, that obviously improves our eradication efficacy. Next slide. So I wanted to show you, you can go to the next slide too, what's happening in the United States. So I mentioned the importance of clarithromycin resistance. In the United States, from 2011 to 2013, this doubled. And so now we're dealing with many H. pylori strains that are resistant to this antibiotic that we commonly include in our antibiotic regimens. Next slide. So Mary Margaret mentioned that, one of the other jobs that I have is I'm the editor of the journal Gastroenterology. And because this is so important, we devoted our very first issue under our term of editorship to focus on identifying the best treatment regimens for H. pylori. And in fact, what these recommendations state is that in individuals who are infected, you should immediately give them 4 different drugs. It can be a combination of a proton pump inhibitor plus 3 other antibiotics, or a proton pump inhibitor plus Pepto-Bismol and 2 additional antibiotics. But whatever you do, treat initially with 4 drugs and treat for 14 days. And that offers the best hope for cure. Now, if a person fails those regimens, we have many other secondary regimens as well. So ultimately, treatment of the infection can be accomplished in over 90% of patients, even though some individuals may have to undergo multiple treatment rounds for this to occur. Next slide. So what about other ways that we can start to eliminate H. pylori in addition to antibiotics? Well, many investigators have tried to look at vaccines targeting H. pylori, and they were not very effective until, next slide, a trial was performed in China 2 years ago, in which they developed a vaccine that was very effective, and they gave this to children, and they were able to determine whether or not they developed new H. pylori infections after they were vaccinated. So on this slide, the columns in green are the children that received the vaccine at 1 year, 2 years, and 3 years, and then in yellow are the individuals who did not. And what they're looking at is the number of new H. pylori infections that these children acquired. And you can see that at every time point, vaccination was very effective in lowering the risk of developing a new H. pylori infection. So I think that there is new hope for H. pylori vaccines. Again, I think that they're going to need to target children because that's the point in time that most individuals actually acquire the infection. Next slide. So what about the future? Well, we hear a lot about personalized medicine or precision medicine. Can we use this to try and identify people that are infected with H. pylori who are at the very highest risk for gastric cancer? Next slide. Well, we've had a great opportunity to work in South America. I mentioned our studies in Colombia. And in Colombia, they have a very unique situation. Almost everybody in the country is infected, but the individuals that live on the coast have a very low rate of gastric cancer, whereas persons that live in the mountains have very high rates. Next slide. So we wanted to do a study to see if we could predict which patients actually developed gastric cancer. And we did this from a genetic standpoint. So we got DNA from all the patients on the coast and in the mountains, and we analyzed the DNA and found that we could categorize different dosing of different genetic elements. And on the pie chart on the left are the genetic results from the coastal populations. They're different from what you see on the right from people that reside in the mountains. Next slide. But the beauty of this study is that we also had the H. pylori strains that came from those patients, those same patients. And so we did a genetic analysis the same way, but we were looking at the bacterial DNA this time. And again, you can see there are differences in the coast H. pylori compared to the mountain H. pylori. Next slide. Next slide. But what happens if we look at these together? So what we found was that if an individual had African background and they were infected with an African type of H. pylori, all they got was mild gastritis. Next slide. If a person had an Amerindian background and harbored an Amerindian H. pylori strain, all they had was mild gastric atrophy. Next slide. But in persons who have a genetic mismatch, that is, their human ancestry is discordant with the type of H. pylori they're infected with, these individuals had the most severe disease. So we think that this opens up many different studies that we can look at and try and identify non-invasively who may be at high risk for gastric cancer. Next slide. The other area for the future that I'll end with has to do with the microbiome. So the microbiome gets a lot of attention now, especially for diseases such as obesity, diabetes, inflammatory bowel disease. What about stomach cancer? Well, these are the results that I showed before from the trial in China, in which people got antibiotics and they had a significantly reduced risk of gastric cancer. Next slide. But they also reported in that study is that when they were followed up 15 years later, about half of those patients still had H. pylori infection, even though they had gotten antibiotics. So you had a paradox. You had individuals that got treatment for H. pylori, but also who had reduced risk of stomach cancer, but they still were infected. Next slide. So this raises another hypothesis in that what are the roles of other bacteria in the stomach that may also contribute to gastric cancer? Next slide. We know probably less about the microbiome in the stomach than what we know about the intestine, but on the left shows the different bacterial species if a person's infected with H. pylori. And H. pylori is shown in green. So when it's present, it is dominant. The pie chart on the right is the bacterial composition of individuals that are not infected with H. pylori, and they're much more diverse. Next slide. We also know that in animals, we can see the same thing. On the left are mice. They have a nice diverse pattern of microbial species. If you treat them with H. pylori infection, to the right you see a much less diverse population. So does this have anything to do with disease? Next slide. Well, in animals, it does. So this was a great study that was published, and what the authors did was they took mice that were germ-free, which is shown in green, so they have no bacteria in their bodies. They're raised in a sterile environment. You can infect those mice with H. pylori alone. 80% of those animals in the left column in green develop no evidence of cancer. And the ones that do, shown in the middle green column, only had low-grade cancer. In contrast, if you infect normal mice that have a normal microbiome with H. pylori, in yellow, 80% of those animals developed high-grade cancer. So we think that there is a collaboration between H. pylori and other members of the gastric microbiota. Next slide. So this is the working model that we have now. We think that early on, after a person's infected, there's a lot of interaction between H. pylori, shown in green, and the host. There's a lot of inflammation. But later on, as acid levels fall, as more bacteria colonize the stomach, it may be those bacteria that actually are important in driving the process late during progression. Next slide. So what I've tried to do today is talk a little bit about the factors that are important in gastric cancer from the bacterial standpoint. And next slide. And how at least work from our lab is, and other labs have tried to put names on all these different factors and specifically how they interact with each other to really fully understand gastric cancer at a molecular level and understand, more importantly, how we can identify biomarkers that can predict disease and identify high-risk populations that we can then go ahead and test and treat. So, Mary Margaret, with that, I'm going to go ahead and end. I'd like to thank you again so much for allowing me to do this, and I'll be delighted to take some questions.
Again, thank you so much for your presentation. This was highly informative about both H. pylori and its treatment, as well as the microbiome. So I'm going to go ahead and start reading off some questions that were submitted during your presentation. If you in the audience still have questions, there's plenty of time to submit them now. The first question I'm going to ask, actually, there's two related questions that came in. So first, should someone with either long-term reflux or PPI use be screened through endoscopy? And in that endoscopy, do cellular changes such as atrophic or non-atrophic gastritis appear as lesions, or would that only be seen in a random biopsy?
So that's a great question. So it's complicated because, you know, H. pylori is also inversely associated with reflux disease, esophageal disease, and esophageal adenocarcinoma. But, you know, my feeling is that if somebody is on proton pump inhibitors for a long period of time for heartburn or reflux, then they should have some type of test to determine whether they're infected with H. pylori. Now, this does not necessarily have to be an endoscopy. You can do a blood test. And if they're H. pylori positive and they're going to be on proton pump inhibitors for a long period of time, then I would definitely treat that individual. Now, if that person is having, you know, reflux symptoms so bad, but to the point that their physician thinks that they need an endoscopy, then during that endoscopy, we can take biopsies from different regions of the stomach to diagnose H. pylori and also determine whether they have gastric atrophy or intestinal metaplasia or a more severe lesion. It's difficult to diagnose atrophic gastritis or intestinal metaplasia just by looking with the endoscope. We need biopsies, and generally that's taking, you know, 2 biopsies from the lower part of the stomach, 2 biopsies from the corpus, and then 1 biopsy from the junction of the antrum and the corpus or the body as well. And then we can diagnose that histologically. What was the second part of the question? Was that it?
I believe that was it regarding, you know, can cellular changes like gastritis or the metaplasia be seen as a lesion versus on microscopy?
Right, microscopy.
So is that, just as a follow-up, are taking biopsies during an endoscopy a standard procedure, or is that something that is, based on the physician's call performing the procedure?
It depends on the indication. So again, there's evidence that, people who have, pain in their stomach, and if you do an endoscopy and you don't see ulcers or you don't see changes that are induced by heartburn or esophagitis, and it looks like a normal endoscopy, still in that situation, I would take biopsies, because there's evidence that in persons that are infected with H. pylori who have what we call non-ulcer dyspepsia, that is pain in the stomach with, what looks like a normal endoscopy. Those individuals, if you treat them for H. pylori, then there is a significant improvement in their symptoms. Not everybody will get better, but it is statistically significant. Individuals that have iron deficiency anemia, if we do an endoscopy in those individuals, then I would take biopsies for H. pylori, because that's a common cause of iron deficiency anemia. But again, it really depends on, the indication for the procedure. There are many upper endoscopies that we do for diseases that, don't really have anything to do with H. pylori. So some patients with cirrhosis of the liver will oftentimes have what we call varices in their esophagus, big bulging veins, and we need to do surveillance on those individuals with endoscopy. Well, they're not having any symptoms that are attributable to H. pylori. We wouldn't do biopsies in that particular situation.
Okay, thank you so much. The next question that came in, and forgive me, I don't know all of the acronyms that were presented, but the question is recurrent SIBO in somebody who's HP negative but has pernicious anemia with IM throughout the stomach?
So SIBO is small intestinal bacterial overgrowth. As far as I know, H. pylori does not contribute to that particular disease. As far as the presence of premalignant lesions, so if a person's diagnosed with intestinal metaplasia or gastric atrophy or dysplasia on biopsy, and for whatever reason the tests that you do for H. pylori are negative, I think that there's still such a strong risk for H. pylori having caused those diseases that I would go ahead and treat that individual with antibiotics. And the reason I say that are, number one, the diagnostic tests that we have are not 100%. They're good, but they're not 100%. Number two, as you develop lesions like gastric atrophy or intestinal metaplasia, you really find that the number of H. pylori in the stomach goes down. And part of that has to do with pH in the stomach. Part of that has to do with the fact that when you talk about intestinal metaplasia, what that means is that the stomach cells are being replaced by intestinal type cells. And H. pylori cannot grow on intestinal type cells. So just because you don't find H. pylori in somebody who has intestinal metaplasia or gastric atrophy or gastric dysplasia doesn't mean that they may not benefit from receiving antibiotics. So I would go ahead and treat those individuals.
Thank you so much. And I know we only have another couple of minutes here, so I'm going to present one last question, which is, how does H. pylori and other bacteria in the microbiome impact treatment of stomach cancer, meaning chemotherapies and immunotherapies?
So H. pylori, many people have, by the time they're diagnosed with gastric cancer, they will have received antibiotics either at the time of diagnosis or shortly after. There's no data, to my knowledge, that H. pylori itself either augments or attenuates chemotherapy for gastric cancer. That's a very interesting point, though, about the gastric microbiota. because there's evidence now, at least in the intestine, that certain bacteria may actually improve the efficacy of chemotherapeutic agents. Right now, because we know less about the stomach microbiota than the intestinal microbiota, I really can't make any definitive recommendations. But I think now, one study I didn't have time to talk about was we've actually looked at the gastric microbiome in those populations in Colombia, the high-risk population and the low-risk, and there are clear differences. There are about 20 bacterial species that are only present in the high-risk or the low-risk populations. And so what we're doing now is we're taking those bacteria one by one and growing them with H. pylori in animal models to see if they increase or reduce the development of gastric cancer. But right now, we just don't have enough information to make strong recommendations about whether a particular stomach microbiota may improve or weaken some of the chemotherapy regimens that are used.
Great, thank you so much. And again, Dr. Peak, thank you for joining us and sharing your expertise with us today. We are unfortunately out of time for questions, but I do encourage all of our listeners to continue to submit your questions by email after the webinar. Thank you so much, Dr. Peak.
Thank you very much. And again, thank you for the honor to do this.
Yes, thank you. And as you know, this webinar was brought to you today by Debbie's Dream Foundation, Curing Stomach Cancer. The recorded version of this and all of our past webinars can be found on the lecture library of our website using the link at the bottom of the slide. I'd like to thank again our sponsors who helped make this webinar possible. Our title sponsors, Boston Biomedical and EMD Serono. Our platinum sponsor, Lilly Oncology. Just as a reminder, please check our website and your calendars to join us for an event near you in the coming months. Thank you so much to all of our listeners today. This concludes our 11th in a series of 12 webinars. Our next webinar and final webinar, will be on January 26th at 12 noon Eastern Time. The topic will be palliative care, the support for those with serious and life-threatening illnesses and their families. Again, please visit our website to review recorded webinars on our lecture library. You can send your feedback, questions, and thoughts by email to patient.resource@debbiesdream.org. Thank you, everyone, for joining us today.